Hi everyone, welcome to ZetaBiology NEET 2020 Quick Revision Series. So today we are revising endomembrane system, and endomembrane system consists of basically four things. First is vacuoles. Okay, so vacuoles are what? Membrane bound space. Membrane bound space. Okay, and its membrane is called tonoplast. Remember this, it is covered with tonoplast. Okay, and what does it do? It facilitates the ion it facilitates the ions against concentration gradients, so it helps in working against concentration gradients. Okay, and third thing is it contains it consists of water, first thing. Second, it consists of cell sap. And third, it contains excretory products, okay, waste products. Excretory products. And so vacuole is a really huge structure, and it occupies around 90% of the volume of cell. Okay, so it's a very big structure. It occupies almost 90% of the uh, cell volume. Other component of this is ER, endoplasmic reticulum. So what does it do? It divides the intercellular space. So intercellular space, it divides intercellular space into two parts. Okay, luminal and extraluminal. Extraluminal. So luminal is present inside ER, and extraluminal, it is present in cytoplasm. This is cytoplasm basically. Okay, now we know that endoplasmic reticulum is of two types. Smooth ER and rough ER. Okay, so smooth reticular is smooth. So smooth ER is the major site of, this is important, lipid synthesis. Okay, remember this. And rough ER, this is the major site of protein synthesis. Okay, yeah. And now the third component. So the third component of endomembrane system is Golgi apparatus. Now Golgi apparatus is a really important organelle, and what it does is packaging. Packaging of material after modification. What else? This is a major site for the formation of glycolipids as well as glycoproteins in cell. The third component of endomembrane system is Golgi apparatus. Okay, and what it does basically is packaging. Packaging of materials after modification. Second thing, it is a major site for the formation of glycolipids and glycoproteins. So remember this. This is an important point. Okay. About the structure of Golgi apparatus is that it consists of flat disc shaped structures which are known as cisternae. Okay, so remember this. And this has basically two phases. Okay, cis phase, trans phase. Okay, and the cis phase is the one that is convex, and trans phase is concave. This is also known as forming phase. Forming phase. This is known as maturing phase. And both of these phases, these are entirely different. These are different, but they are interconnected. Remember this. These are interconnected. So thanks for watching.